Thanks everyone for coming here today. I recognize this is over your lunch break, so I appreciate that you're taking some time out to come talk sequencing and be microbiology nerds along with me. Also, I should be thanking Jason, who invited me to this education series. It's really cool to be able to have these bridges between the clinic and the hospital as well as the university. My name is Keith, as you can see on that slide, and I'm a postdoctoral fellow at the University of Regina. So I work with Dr. Andrew Cameron, and the Cameron Lab is focused on using sequencing technologies as a way to study microbes. And it's really an exciting era for molecular microbiologists such as myself because of how rapidly changing sequencing technologies are changing basically how we approach microbiology research altogether. So my goal for this talk today is I'm going to share with you some of the latest sequencing technology developments. I'm going to talk very specifically about the field of whole genome sequencing and how this concept or this idea of whole genome sequencing is emerging as an important tool in infectious disease control. So to start off, it's no secret that antimicrobial resistance is an incredible threat that we are currently dealing with on a daily basis, even today. But it, of course, poses an even greater threat to the future if we have inaction about it. So many countries are starting to sign on to this fact, the idea that we really must start addressing antimicrobial resistance and having better stewardship of those antibiotics that are available to us now. So an example of this is coming from the UK, and in 2014, the then Prime Minister, David Cameron, was asked to commission an economist, of all people, to talk about antimicrobial resistance. And so he asked this economist, Jim O'Neill, to have a look at the problem from two aspects. First of all, what's going to be the total impact of antimicrobial resistance on the future in terms of human health as well as the economy? And then the second aspect he wanted him to look at was to come up with some concrete action plan, the idea that we have some ways that we can deal with resistances. So the report came out about 3 years later, and in it was this very doom and gloom statement, the idea that today's already large 700,000 deaths that occur every year that are attributed to antimicrobial resistance would become an extremely disturbing 10 million every year by the year 2050, which sometimes sounds really far away, but really isn't that far away in the grand scheme of things. And to put this into context, he compared this to cancer, saying that this 10 million people and these 10 million mortalities would be more people dying to antimicrobial resistance than those that die to cancer, which is around 8.2 million people that are dying every year right now. So it's very obvious by all of these numbers and these estimates that antimicrobial resistance is something we need to address. Now, the silver lining around this doom and gloom quote that I've put up here is that this is an estimate that is taking place if we do nothing about antimicrobial resistance. So, of course, the other part of this report is challenging us to look at what ways we can do action on antimicrobial resistance, how we can address this problem. And some of these things you see in your everyday practice. Public awareness programs that are going on that are making it so that the public is aware that drug resistance is a problem and that it affects all of us. So this is one of the recommendations in the O'Neill report. We need to be pushing these public awareness campaigns. And one of these includes Choosing Wisely Canada, which Jason talked about a few months ago here in the same education series. And this particular campaign is very useful because it includes information for both the public and helping them understand when they should be looking for antibiotics as part of treatment for their illness, as well as it includes tools for practitioners in the healthcare settings so that they can do things like having these viral prescription pads and help patients understand how they can make themselves feel better in lieu of getting antibiotics. So overall, the message, I should go back actually. The other part of this message is a bit closer to home. So one of the other action points that were shown up in this report is this idea that we need to address antimicrobial uses also happening in veterinarian practice as well as agricultural production. So I'm originally from Saskatchewan. I'm from a small farm on the southwest corner. And I can very much remember going to the city on the weekends, going to PV Mart and being able to grab an antibiotic off the shelf with Dad and bring that home, and we would use that to treat our cattle when they were ill. Well, as of December 1st of 2018, Health Canada has made it so that we are moving away from this practice. So no longer can you go to PV Mart to grab antibiotics for your animal. Instead, you're going to have to develop a good rapport with your veterinarian, and that veterinarian will help you and decide when it's time to prescribe an antibiotic for that illness. So overall, the idea that we need to increase our stewardship of those antibiotic agents that we have in our current arsenal, because they are a limited public good. So every time we go ahead and do a prescription, that may be taking one prescription away from a future illness. So the other part of stewardship, apart from trying to reduce this sort of excess or unnecessary prescribing practice, is the need to be able to decide when is it a good time to be prescribing. So this other quote is one that I pulled out from the O'Neill report, where he says, "I find it incredible that doctors must still prescribe antibiotics based only on the immediate assessment of a patient's symptoms, just like they used to when antibiotics first entered common use in the 1950s. Now, this might be a bit generalized. I'm not going to say that I know necessarily because I'm not a clinical microbiologist or a medical doctor. We do have some diagnostic techniques that are available to us. And for some of these pathogens, those diagnostics are very rapid, they're inexpensive, they're able to help with prescribing practice. But that's not necessarily true for all pathogens. Some are really hard to grow by culturing techniques, which have been around since the 1860s. So what the statement from Jim O'Neill is saying is that we really need to start looking at diagnostics. How can we be helping healthcare practitioners to make better choices and be able to have all the information they need to prescribe effectively when they do? So as I was saying at the start, I'm a molecular microbiologist. I'm not a clinical microbiologist by any means. So when I was approached with this project of looking at an outbreak of disease, I really needed to orient myself to how is it that hospitals control infectious disease, because I had no idea at all. So this is an example that comes from the United Kingdom of what their process for controlling infectious disease in hospital settings looks like. So some of the first steps include surveillance. So patients that are constantly being screened for the organism and its presence once they test positive once. And then if there's incidences where there's 2 or more patients that are testing positive for the same organism, this can act as a trigger to initiate an outbreak investigation, epidemiology investigation into that outbreak. And of course, the main focus of this outbreak is trying to figure out if there's any links between those cases. So some of the common descriptive epidemiology that we might be using is looking at time and place and seeing if there's any cases where those patients were overlapping during their stay in the facility, and whether or not we could link them by, say, those factors or other factors, say, if they had shared healthcare practitioner teams, for example. And so hospital databases become a really important part of this, keeping all this type of metadata that can help us to track disease and infection in a hospital setting. So once all that information is collected, the infection control team sits down and they make a decision as whether or not they need to investigate further. So this can involve screening additional patients that haven't tested positive yet, potentially screening some healthcare practitioners in the facility, as well as screening equipment and the environment itself. So broadening their investigation. And they may also act through interventions to control that spread, including maybe some changes to some of the cleaning regimens or ways that healthcare practitioners are interacting with their patients. So what I found very interesting as a microbiologist is where bacterial typing comes in this whole idea of trying to link infectious diseases together. And so what I found, at least from this paper and this particular report, is that oftentimes molecular typing, it can be used, but most times bacteria are related based on their antibiotic resistance profiles. So if they show resistances to similar antibiotics, this is often enough in this investigation to suggest that, yeah, maybe these cases could be linked. So that's kind of a more routine, I guess, approach to it. Again, I'm not necessarily sure that that's always the case, but there are molecular typing tools that can help to understand bacteria and their relationship and help with these linking of cases if there happens to be a link there. So there are a number of different types of molecular typing that exist out there, and I'm going to focus specifically on some of those DNA-based methods that we are currently using to type our bacteria and look for these relationships or links in cases. So I won't go specifically into how each of these technologies work, but the general idea is that DNA-based methods generally will sample only a fraction of the total genome. So you can see on the screen that there's that circle, and that's to represent the bacterial genome. So in this case, that would be like the bacterial chromosome that you would get from that bacteria. And you can see based on the colors from these different tests, that different tests will be testing specific regions within that chromosome, looking to see how that sequence compares from one isolate to the next. And by doing that comparison, you can determine if those cases or those bacteria are potentially linked together, that they may be the same bacterial type, or maybe they're completely different altogether. So one of the shortcomings that comes up with some of these cases is that they don't provide the resolution that we need all of the time. So MLST, for example, multi-locus sequence typing, as well as RFLP, restriction fragment length polymorphism testing, these are things that we are constantly using still even to this day in epidemiological investigations, and they are very effective in those investigations. But when it comes to the hospital settings, sometimes they don't provide the information and that resolution that we need. For example, oftentimes we'll get the same sequence type infecting a hospital multiple times, but that doesn't mean that all of those infections are related just because they share a same sequencing type. It could be that they share sort of similar sequence at those specific locations that we're sampling, but the rest of their genome is in fact very distinct from each other, and it makes it so that they're completely unrelated bacterial isolates and unrelated cases in the hospital. So this is maybe one of those shortcomings that come from our current molecular typing schemes and why they aren't implemented as regularly as we maybe think they should be. So why is it that we are only sampling parts of the genome? Why is it that we limit our typing to these particular fragments? Well, the reason for this really is based on the history of how we do DNA sequencing. So Sanger sequencing has been around for a very long time at this point. It was developed in 1977 and really forms the core basis of all of the sequencing that we do routinely in the lab, be it if we're doing genetic cloning or some of these molecular typing methods that we do for hospital outbreaks. These technologies, these Sanger sequencing technologies, were also used in some of the biggest achievements that were accomplished in the 1990s and the early 2000s. So for example, we use these sequencing technologies to assemble the first bacterial genome. So this would be a whole genome sequence, which I'm going to talk more about here today. And the first one was actually a very small pathogen in terms of its chromosomal size. So it's 1,800 million base pairs. And that will become more important later on here. We also use Sanger sequencing for the Human Genome Project. And for those of us who can remember that project, it was a massive undertaking that involved these rooms like the picture on the left that had many, many sequencers all sequencing human genomic DNA at the same time. And even though we had this massive capacity, it still took 15 years to be able to sequence and assemble that whole genome. So based on that, based on the fact that it took so long, it became recognized by scientists that there's maybe some shortcomings in the way that we are doing sequencing up to this date, and maybe there's some ways we can be innovating these technologies. And so shortly after the release of the Human Genome Project, we start to see the rise of what are called these next generation sequencing platforms. So these are platforms that are very efficient, much smaller, and much higher power and throughput in terms of the amount that they can sequence. So how exactly does this work? How has genome sequencing changed since the 70s? So I'm going to use the Haemophilus influenza project as an example of how this works. So one of the challenges that scientists had encountered when they were doing this whole genome sequencing was the fact that Sanger sequencing is limited to sequencing about 500 base pairs of DNA within a sequence reaction. And so you have to take this shotgun approach where you take the genome, which is much larger than 500 base pairs, break it up into these smaller fragments, and then sequence each of those fragments individually in separate reactions. So this is why Sanger sequencing became a really intensive process for when it came to whole genome sequencing. The next generation sequencing platforms such as Illumina have changed this game altogether by making it so that instead of having to set up all these separate reactions, we can do it all at once in parallel. So you can see in the picture on the bottom here that there's a couple of colored dots. And so what that represents is basically each of those colored dots would be a reaction that's happening in one of those tubes. So we can more or less put tons of Eppendorf tubes onto what looks like a slide and be able to sequence multiple fragments all at once. And this accelerates our ability to get all the fragments, all that genome sequence that we need to then reconstruct the genome of interest, much like you would a puzzle with puzzle pieces. So on that same metaphor, sometimes putting these puzzle pieces together are not easy. These small fragments can share a lost sequence identity, and sometimes we can line them up correctly using computers to reassemble that original genome that existed in that bacteria. But sometimes the computers get very confused because some of these sequences look alike and they may not actually belong together. So a much newer technology that has come out called nanopore sequencing has been really important in our ability to reassemble these puzzles that are bacterial genomes. And why that is because nanopore can do much longer sequences. So rather than being limited to these 500 base pair reactions, we can actually go up to, in this case, 2 million base pairs, which if you remember from the previous slide, that's almost equivalent to that Haemophilus influenza genome that they were trying to sequence. So we can suddenly be doing a puzzle that only contains 10 pieces as opposed to 1,000. And that makes it much, much easier for us to assemble these whole genomes. So now that we can be doing this whole genome sequencing and assembling the total sequence that's available in all these different clinical isolates that we're acquiring, we can start to look at new fields and new ways that we can apply this to infectious disease. And one of the ways that we do this is through genomic epidemiology. So what is genomic epidemiology? Well, as the name suggests, it's kind of combining this whole genome sequencing that we do with classical epidemiology that would be used in these outbreaks. So the principle of it is that on the left, you have DNA sequencing of your pathogen. And as you collect all those sequences that come from different isolates, we can look at how they're the same and how they're related, but we can also look at their mutations. And mutations give us a sense of time and chronology. So as bacteria continue to evolve over time, they start to collect more and more mutations. And so that allows us to look at cases, say 2 isolates coming from 2 separate patients, and one patient that maybe comes later on will have a genome that contains more mutations than the patient that originally was infected. So we can use those mutations as sort of a biological clock, looking at the chain of transmission between patients. We can then use our descriptive epidemiology to overlay metadata about space and time, and that can help us to really determine the relationship between these cases and get down to the big question of what are the source of some of these contaminations, where are some reservoirs in the hospital that are causing these patients to get sick. And so ultimately, when we combine these 2 approaches, we can get this ability to track the spread of disease. So that's maybe a bit abstract. So I thought that I'd maybe pull out an example that I like to use in some of the classes I've been teaching. So I'm sure many of you are familiar with telephone, or if you're not, I'll give you a quick rundown of how this game works. So you often would have a room of people, and they'd be standing in a line. And somebody would be at the start of this line, and they're going to choose the word. So this is their word that they're super secret about. And they're going to pass it on to the next person, the person that's closest to them, and whisper it in their ear. So in this case, we're looking at the word please. And that person's going to hear that word and try to pass on the same thing to the next person. But part of the joy of the telephone is that oftentimes these words start to get confused. And so you get similar sounding words getting passed down this chain of people to the point that once you hit the end of the chain, you see a word like fees that looks kind of similar to the original word, but it has some mutations. It's kind of changed during this transmission through all these different people. So really, you can start to think of sequencing in the same way, where really sequence just becomes a word. And as that word and that sequence and those isolates get passed from person to person, you can start to see that mutations are happening in that word or sequence. And so those mutations can tell us about who's most related to who, or who is standing next to who in that transmission line, versus later down the road where you have something completely different than what's at the start, and probably unrelated cases. All right, so that's kind of my example of how genome sequencing can be applied, how we're using this in infectious disease. And so now I'm going to talk about a real-life example of how we've been using this here in Saskatchewan to address a local problem. So vancomycin-resistant enterococci is indeed not something just in Saskatchewan, something that globally we are dealing with in healthcare. And it's one of these indicator organisms that we're constantly surveilling for and making sure aren't causing outbreaks in our hospitals. So part of the reason why enterococci are so difficult is the fact that they are just a normal colonizer of healthy people. So you can find it in about 19 out of 20 people. And just because they are colonized doesn't mean that they are infected. However, you can have cases in more sensitive wards where people may be immunosuppressed, they may have barrier breaches such as catheters, or they may have undergone previous antimicrobial treatments. And these things can put them at risk for what normally would just be a colonizing bacteria to start causing infection by getting into the wrong places in the body. The other really difficult component of enterococci, especially in this hospital environment, is that they can persist for a very long time. So there's some reports in terms of how long they can persist. And one of the estimates is the idea that they can live for 5 days to up to 4 months on different dry inanimate surfaces. So it's really important that we be able to control this through good cleaning and good hygiene in our hospital setting. So this is just a diagram to illustrate some of those routes by which enterococci can be moved from, say, someone who is colonized with that pathogen. So it can move through contaminated equipment, say contaminated hands of healthcare personnel, as well as contaminated surfaces that can be found throughout, say, a ward. And that once a susceptible patient comes into contact with any of those contaminated surfaces, that can potentially cause an infection. So our lab was involved with an investigation. This is a retrospective investigation, so that means that it was an outbreak of disease that had happened previously. So in this case, it was happening in 2013. And what was happening is we were having lots of VRE cases. Patients were testing positive, either colonized or infected, with VRE during a stay in a single ward in a hospital. So this was occurring over a 9-month period of time. And the reason why we did a much further investigation and research into this was because it involved implementation of more extensive control efforts. And despite the fact that we were changing and trying to manage this infection and control the outbreak, it continued to persist within this ward. So the infection disease control team had come to us with 2 major questions. Was the ward continuously contaminated over these 9 months, and was this one single outbreak where we weren't able to find all the reservoirs of this pathogen? Or was it that VRE was introduced into the ward multiple times, potentially from other wards or from the community itself? So we started to do this whole telephone genomic epidemiology technique. And in order to do that, we really need to have a picture of the puzzle. So that means that we're going to create our reference genome in which we're going to then compare all of the sequences we get from all of these different clinical isolates and look at these differences or these mutations. And what you can see here is that with the short read sequencing that Illumina provides, that sometimes our ability to rebuild or reassemble that puzzle, which should look like a circle, sometimes it doesn't turn out that way. And it's because the computer has a really hard time discerning how these sequences should go back together again. So it's only with these new longer read sequencing technologies, or if you have an available reference genome that's been previously sequenced very intensively, that you can start to get a much clearer puzzle picture where you get these completed bacterial genomes that all look like circles. So the big circle here would be the bacterial chromosome, and then all these other circles would represent plasmids or mobile elements that can pass in between all these different bacteria. So having that reference genome in place, we started to investigate the sequence relatedness of all these isolates, and we overlaid some epidemiological metadata on that to see if we could track transmission. So the outbreak had started in March. And what we see is that there are about 16 cases where we have bacteria that look genetically identical. And so they form what looks like this node. And so that tells us that these cases are highly related, that all of these people are likely infected with the exact same version of VRE. And so we can start to apply epidemiological metadata to this. And what we find is that a lot of these cases involved patients that were staying in the same bed or in the same room as someone else that had been VRE positive. And in some cases, we were able to see that there would be patients that were sitting in adjacent rooms and that they shared healthcare professional teams. Later on in the infection, or in the outbreak rather, in April, we start to see this large number of cases, so 24 cases. And there's a lot of things going on with this, so I'm going to draw your attention to 2 major things. So the first thing is that there's all of these branches. So these branches represent mutations that are happening in isolates between one patient to the next. And so that allows us to establish these branches that show us this chain of transmission. Now, what you'll notice is that some of these branches are directly connected to that red node. And so what that tells us, or what we can infer, is that there was probably some contamination that had remained from that starting of the outbreak. And so perhaps there is a reservoir that had been missed during cleaning. The other thing is that you can see that there are multiple branches going on here. And that means that we have multiple different transmissions happening in parallel. It's not the case that all of these patients were related together and all of them were passing around. It was the case that some patients were sharing it with other patients. Meanwhile, there might be a different cohort of patients that were sharing it with each other as well at the same time. We then move on into September, where we started to see, again, a large number of genetically identical VRE. So in this case, we see 11 VRE positive cases where the VRE itself shares an identical genome at that big blue node. And what I want to draw your attention to again is that any of these regions in blue, you see that they directly connect to these red and green nodes. And what that tells us is that, again, there must have been some sort of contamination or source or reservoir somewhere within the ward that perhaps had been missed during cleaning or something like this. So what this suggests altogether, both the short branch lengths and this connection to this big red node, is that perhaps this was one big outbreak and that it was something that was happening in the ward and continued for these 9 months. So there's many other things I'd love to talk to you about. I'm going to breeze through them really fast here at the end. But I focused a lot on sequences that are similar between all these bacterial isolates. So that's considered these sort of conserved sequences that allow us to compare these isolates directly. But there's much more to this picture. So whole genome sequencing also allows us to look at accessory sequences, so sequences that may be unique to different isolates or different bacteria. And a lot of these accessory sequences are really important in healthcare settings. They can lead bacteria to acquire new resistances to antimicrobial reagents. They can become more virulent. They can persist for longer based on the genes that are carried in these elements. So we wondered whether or not we see some of these genetic elements that may have contributed to the outbreak. And what we see as we go back to our picture of that genome that we built is that there was an 88,000 base pair plasmid, so this mobile element that can be shared between bacteria, that started to pop up in cases about mid-outbreak. And so we're going to continue to try and characterize this plasmid and figure out what maybe it could have contributed to the overall amount of VRE cases. So is Regina unique in this outbreak? That was one of our major questions as well. So we can go back to some of our classic or more long-term standing methods, such as multi-locus sequence typing, to do a comparison of how VRE in Regina relates to the rest of the world. So we see that Regina carries the ST80 version of VRE, sequence type 80. And if you probe the literature, you can find that, in fact, many countries were dealing with ST80 outbreaks at the same time. So in Regina, this was happening in 2013. However, in the world, it was happening in cases between 2014 as well as 2017. And what's great about this is that many of these countries are also adopting whole genome sequencing technologies and approaches. And what this means is that we can have these big databases and we can look at global transmission. How are these pathogens moving, not only within hospitals, but maybe within countries as well? So accessory elements and all of these things about whole genome sequencing are obviously very important for how we can track infectious disease. But there's many things that we can look at and expand upon past these sort of retrospective studies that we can do with epidemiology. So one of the things that is making it so that whole genome sequencing really can apply to infectious disease control today is the fact that we can start to have culture-free pathogen detection. So I mentioned that a lot of pathogens have to be cultured, and this sometimes works for some pathogens, but not for others. And what we're seeing are these new technologies called DNA capture, which use these probes. Basically, they're also sequences that are matching to the genome sequences in our pathogens of interest. And so what we can do is take these probes and add them to primary samples, say like fecal cultures that we're getting from patients, and pull out the DNA of the pathogen if it happens to be there. And so what this does is this really rapidly accelerates our ability to diagnose whether or not a patient is carrying that particular bug. So this is an example of something that's already on the market. It's called VIR-CAP-seq, and this is a capture panel that's been developed. And so it says here that the VIR-CAP-seq capture panel consists of genomes of 207 viral taxa that are known to infect invertebrates, including humans, and enables detection of viral sequences in complex sample types. So really, we can start to do this massive panel or screen looking at whatever viruses or bacteria are important to us at this time. And what this means is as we pull this DNA sequence out, that we start to have greater sensitivity. So we can detect infection or detect the existence of these pathogens within patients much earlier and earlier. So for example, they say that it results in a 100 to 1,000-fold increase in on-target viral reads versus if we just tested for the presence of that pathogen directly from a fecal sample, for example. So there's other things, of course, that having this whole genome sequence is going to help us understand, both from a research perspective as well as a sort of predictive perspective of whether or not an infection could go wrong. And one of the ways that many institutions in the globe are trying to make this work is by developing these sorts of genotypic panels for antimicrobial resistance phenotype prediction. So can we use the DNA to figure out whether or not this pathogen is resistant to different antimicrobials we might use otherwise? So this is something that is actively being developed and actively being used. And you can also imagine that we can look at, say, risk prediction. So are those VRE that we're finding in colonized patients, do they have a potential to gain new virulences that could lead colonization into infection in that patient? The last one I want to touch on is this idea that we can enable other preventative approaches. So diagnostics, of course, is an important tool in antimicrobial stewardship, but we need more than that in terms of innovation to battle this problem. And one of the ways that we can do this is through vaccines. And so one way that we can integrate whole genome sequencing and that technology along with vaccine is this method called reverse vaccinology, where we take these whole genome sequences and identify genes and sequences present in these pathogens that are going to elicit a good immune response from a person. And so this is actually something that's being used at home here in Saskatchewan. This is an example from Vito, which is where I did my PhD. And they're using this reverse vaccinology approach to inform their vaccine design against mycobacterial diseases in cattle. So in summary, I'm hoping that I convinced you today that sequencing technologies have vastly improved in speed as well as throughput and efficiency and the amount of information that we can collect. And that pathogen characterization via whole genome sequencing offers important insights into how we can approach infection control. And ultimately, the whole genome sequencing may be this holistic approach that we could be using in antimicrobial stewardship, helping us not only to control disease, but also develop additional technologies such as DNA capture or vaccines that are helpful in combating disease. So with that, I'm going to end on my acknowledgement slides. I would like to specifically shout out to my supervisor, Dr. Andrew Cameron, as well as to Dr. Jessica Minion, as well as Dr. Meredith Ferris, who are involved in this study. As well to one of our collaborators, Dr. John Van Ham, who's out in BC, who helped us with this nanopore sequencing of our isolates. Members of the Cameron Lab who are here today to watch this talk, as well as those who are involved in the study, as well as members of the Yoast Lab who are also here today. Thanks for being here, you guys. And I'm happy to take any questions.
If there are any questions in the WebEx world, please unmute your mic and go ahead and ask or type in your questions.
I have a question. You mentioned at the beginning policy changes regarding antibiotics in veterinary use.
Yes.
Are you aware of any policy changes that have happened or are in the process of happening on the human side for how antibiotics are used or regulated?
Oh, interesting question. I'm not personally aware, so I think a lot of the attention has been on this veterinary side because they are such large changing rules, but I'm not personally aware of how prescribing practices are maybe changing in the healthcare setting, so I need some of these healthcare individuals to help me out on that.
I can take a stab at that. So it's Jason in Regina, everyone, if you're listening in there. Policy-wise, so on the federal government scale, there are action plans and things like that that are being made to start addressing some of the shortcomings that come in the world of antimicrobial stewardship, so getting better surveillance and those kinds of things so we can better understand the scope of the problem. Actually putting those actions into action really falls to the level of local antimicrobial stewardship programs and health authorities and health regions and things like that. And so that is a much harder piece to sort of get going. It requires people and resources to put that information and to do the sort of knowledge translation to get the change at the clinician, the level of the clinicians. So policy-wise, there's not a lot going on. Of course, there are, with the way pharmacies are set up and the way pharmaceuticals are set up in the human world, it's much different than it's been in the animal world. But yeah, so it's a little more, I hesitate to say advanced, but it's different there. But in terms of actually putting some of that stuff into practice about actual appropriate use of antibiotics, no, it's still got a ways to go.
So,
Thank you.
so for the vancomycin-resistant enterococci, so you have an idea of how many resistances it carried? Was it multi-drug resistant? So,
yeah, so of course this is something we can be asking now that we have those whole genome sequences. We haven't gotten that far in this study.
Okay.
But it's something that we certainly can do. Again, this idea that we have the whole genome, we can look at DNA, and that there are really well-curated databases that include all these antibiotic resistance gene sequences. So we can really just pump in our completed puzzle, our completed genome, and have a look at what antibiotic resistances in terms of genes are potentially there.
Also interesting that he had a plasmid, so probably could those plasmids be kind of sources for virulence genes? Or could it be, or they were like SPR-like islands too? So what do you think could have been? What do you think could have been responsible for the movement of the virulence genes into that particular bacterium?
Right. Yeah. No. Good question. I'm hesitant to say what it is that this plasmid is doing, whether or not it's conferring additional resistances or valences. Again, it's something that we haven't completely characterized past using just that base genome sequences to look at sort of the relatedness of all of these isolates. So it's very exciting. It'll be neat to see what genes are there and potentially if they've impacted the way that these infections or colonizations were occurring.
Hey, Keith.
Hey.
So you mentioned using DNA capture as a rapid sort of clinical tool for diagnostics directly from a sample to avoid culturing. But I know that DNA capture is somewhat expensive right now.
Yes.
Do you think that there will be a similar trend with DNA sequencing where it will become cheaper over the next 10, 20 years, where it's actually feasible for hospitals to use DNA capture as a diagnostic tool?
Yeah, great question, actually. Yeah, so I mean, this is something that we are even using in our lab, as Danae has mentioned, that we're in fact looking at DNA capture as an approach to do some of the studies that we do. Whether or not it's going to become less expensive over time, I would say that I could predict this following the same path. So, you know, the idea that with Sanger sequencing, it was working for what we were doing at the time. And then once we started to really challenge ourselves, can we build these bigger genomes, such as the human genome? That's when we started to recognize some of the limitations that were available in our current technology. And that's when next generation sequencing was born, really was out of this human genome project and the challenges there. So to answer your question more directly, I would say that DNA capture is expensive now. It's a relatively new phenomenon. I believe it came around 2014, 2015. I could be wrong on that VirCap-seq that's available on the market. But that's one of the very few examples of DNA capture and its applicability right now. So I think that as we start to explore this more, the same phenomenon is going to occur where maybe it becomes less expensive to manufacture those probes that we can then use in this technology. Oh, Jason.
So there was a couple of your slides where you were looking at the spread of and the changes in the bacteria, and so you know you're talking about reservoirs in the that might have been missed during cleaning and stuff like that. But I noticed a shared healthcare worker popped up in two of the periods there that you were looking at.
Right.
So is that something that's just I'm assuming that's based on just you guys are looking at the whole thing and saying, okay, there was a shared healthcare worker, so that could be a potential source of sharing the infection, I suppose, right?
Absolutely right. Yes.
Okay. I guess it's not really a question then. I just think that's a great point for a clinical group where those people are, because it is, showing that you could be one of the factors that is contributing to this. And so just that importance, pointing, to the importance of hand washing and proper PPE use and that kind of stuff. So then the other question is, the question I guess then is, will you guys be able to look at, or are you planning on looking at, and it was already sort of brought up, the different types of resistance. And so I know one of the things that is popping up with VRE, there was a study, I think from last summer, that looked at resistance in VRE to actually alcohol, killing by alcohol in places in Australia.
Totally.
So is that something that you guys would plan on looking at as well, instead of just your typical resistance genes and stuff?
Yeah, no, great question. Personally, I'd love to. So my history in terms of my PhD was looking at survival of salmonella. And so I would love to look at survival and persistence. I think that's completely relevant. If doesn't matter how diligent healthcare professionals end up being, if their cleaning practices aren't working for them, they can try as hard as they might, but that pathogens can still exist in these reservoirs. So that's one of these hazards that you have to take when we're looking at this descriptive epidemiology, is that just because there is that pattern, it doesn't necessarily mean that's exactly what's happening. But again, to answer your question in terms of like alcohol resistance, yeah, hugely important. I think that we really need to expand our idea past looking at only antimicrobial resistance, only virulence genes that can influence infection. Really, let's look at how are these pathogens surviving and able to transmit for potentially even 9 months in this case.
Just piggybacking a little bit on what Jason had to say. So we found out that, okay, there was likely a reservoir, something had happened here. What was then the communication to that unit, and what actions happened afterwards to help improve this problem now that we had somewhat of an answer as to where the VRE was coming from or how it was being continued to be spread?
Yeah, no, very good question. So at this point, this is very preliminary. So what I'm presenting to you, I don't think those professionals actually know this information yet. And so what this is going to take is doing more interpretation of this data. And unfortunately, that's one of the downsides of these sort of retrospective studies, is that trying to find those smoking guns, trying to find exactly how we can change our infection control, it becomes a little more difficult, right? So, but yeah, no, certainly this is still a study in the works. What I present to you is very preliminary even at this point. Yeah.
All right. Well, if there are no more questions, I would like to thank you once again, Keith, for your wonderful presentation.